cell physiology, concepts that you need to know for anatomy and physiology that come out of cell physiology. I'm going to do this breathtakingly fast because it's review. And it won't be comprehensive because it's review. But there are some things that I want to make sure we do in this room for this class. So we're going to focus today on ion transport, distribution of ions and solute transport. Then on Tuesday, we'll do some receptors and electrophysiology. So this part of this class and the next class will be key concept review from Cell Phys. So here's a slide that you saw from Cell Phys of the 4 types of transport across biological membranes. Simple diffusion, where the solute makes contact with lipid and because of its amphipathic nature can dissolve in both water and in lipid and move through cell membranes. This is going to be the least important of the solute transport mechanisms for this course. It's pretty important for the other AMP course, but it's not very important for this semester. Another form of passive transport is through ion channels. Actually, the channels, most of them are ion channels, some of them are water channels. Those are the 2 types of channels that we focus on. The solute, the ion, typically does not make contact with lipid. It interacts with the protein, the channel, but it flows down its concentration gradient passively when the channel is open and doesn't when the channel is closed. There is going to be a transporter-mediated passive transport. Uniport is what we called it in Cell Phys, where the solute interacts with the transporter and the transporter makes a conformational change for every single time the solute moves. I think I've got a clicker question that's going to ask you. I'm going to reinforce that information in a clicker in a few seconds. Then we have active transport. Remember, there's primary active transport, where the transporter uses ATP to transport the solutes. And there is secondary active transport, where one gradient provides the energy to pump another solute against or up its concentration gradient. These are concepts that are a review from Cell Phys. The textbook covers it well, so if this is not feeling like review, as if this is feeling like, where is this coming from? Use the textbook, either your Cell Phys textbook or the AMP textbook. So let's do a clicker. You know the drill. The clickers are over here if you didn't pick one up. We'll do the same thing that we did in Cell Phys last year. Okay, by the way, as long as we're doing this right now, the syllabus, the schedule, key concepts for this section, the picker, and a printout of today's PowerPoint. Not all slides, but key slides are in piles over here. So make sure, let's take a little break right now and make sure everybody has everything you need. Okay, so first clicker of 2020. I think. I'll just be honest, what are the main differences between simple diffusion and channel-mediated passive diffusion? These are both passive transport. We're talking about the differences between simple diffusion and channel mediated passive transport. Show me your answers, please. Okay, so let's go through it. Is A a true statement? No. Somebody who didn't choose A, explain why A is a false statement.
Channel mediated transport also requires
Absolutely. They're both passive, they both require a concentration gradient for there to be any meaningful flux by either mechanism. Okay, in simple diffusion, the solute contacts lipid, but in channel-mediated transport, the solute contacts protein. Is that a true statement? Yes, it is. Lipid, protein. Simple diffusion happens without protein conformational changes, but most channel-mediated transporters, ion channels, undergo protein conformational changes. Is that a true statement? Yes, it is. This is showing an open channel, but channels have gates. They open and close. The opening and closing of channels is a conformational change. Most channels undergo conformational changes in response to voltage changes, in response to ligand binding, even if they are constitutively active. That doesn't mean they're open all the time. They could be just transitioning between opening and closing with no stimulus to change those kinetic changes. Conformational changes are happening here and not here. Channel-mediated transporters are selective for one or a few solutes. Is that a true statement? It is. Most ion channels have selectivity filters to allow them to choose, to select between one or a few types of solutes. So the best answer was B, C, and D. A was the only one that was wrong. So most of you got partial credit. Okay, let's do another one. So again, remember the point of these clickers is not, let me go back. The point of these clickers is not for me to test you. This is not assessment. This is to initiate a conversation, which I didn't really allow. So that's why we're going back. Are there questions about this material? That, you got something wrong. Do you want to follow up with, wait, I thought da 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 da, or could you explain why da 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 da? That's what these are about. So if I move too fast, like what I just did, please just raise your hand and say, wait, I don't understand why D is true and A is false. I know in a room this large with this many people in it, there's a high energy barrier to raising your hand. I understand. So feel free to ask Brittany a question after class or me a question after class. But if you have the ATP, raise your hand and ask the question, because everybody will probably benefit from your question. I'll be honest, what are similarities between channel-mediated passive transport, ion channels, and transporter-mediated passive transporters, uniporters? So what we're talking about now is we are comparing channel and uniport. Okay, go ahead and do the clicker and show me your answers. And now we're talking about similarities, not differences. Show me your answers. Okay, so I'm seeing mostly fully correct answers on this one. So this is an example of where I saw most of the class got it most of the class got it completely correct. A few people got it partially correct. Again, it doesn't matter really whether you got it fully or partially correct now. But now is an opportunity to raise a question that could be the benefit for the other people that got it partially correct. So are there any questions about why these are all true statements? Okay, let's do another one. Question?
I didn't know why A was correct. I didn't know that.
Okay, why is A a true statement? The only way for there to get to be flow or flux through one of these transporters is for there to be a concentration gradient, because they're both passive. They're both passive, so the only way for an ion to get meaningful flow through a channel or a uniporter is to flow down their concentration gradient. The only way to get a concentration gradient is to have it previously established. These transporters are passive. They don't establish gradients. This is a good point. They don't establish gradients. They use gradients that were established by active transporters. Remy, did that help?
Yes, thank you.
Other questions? Yes?
Can you go over what channels go through binding?
So it's clear from your question that you understand that in the uniporter there is a interaction here, a binding and then a conformational change and then unbinding. In channels, the act of selectivity requires at least something like binding. There is an interaction between the solute and a selectivity filter, a selectivity component of the channel. And that interaction is high enough affinity because it's selecting for sodium over potassium or for chloride over potassium or something. It's having to select, and that selectivity comes from selective binding. The kinetics of that binding are real different, but they're still binding. Other questions? Okay, let's do another one. Primary and secondary active transport. So now we're going to active transport. We're comparing primary and secondary active transport and which are the true statements. So we transitioned from passive to active, and now we're comparing primary and secondary active transport. Okay, I'm seeing a lot of partially correct answers again. A, let's do this one at a time because not everybody got it right. A, is A a true statement? No, somebody who's shaking their head, tell us why.
Secondary active transporters don't use ATP, they use like a different solute going down its concentration.
So primary active transporters use ATP. They hydrolyze ATP, but secondary transporters don't. So that's what makes A false. B, all active transporters can create concentration gradients. Yeah, that's what they do. B is a true statement. Either they're using the energy from ATP to create a solute gradient, or they're using one solute gradient to create another solute gradient. Even in the absence of energy input, the reaction catalyzed by active transporters is spontaneous or favorable. Is that true? No, that's false. Creating a gradient is not favorable. Things going down their concentration gradient is spontaneous and favorable, but things being pumped to create the gradient is not favorable unless there is energy input in the form of ATP or another solute gradient. So that's why C is a false statement. Is D a true statement? Active transporters are a type of enzyme. I can see, this is a fuzzy one. I think it is a true statement. Primary active transporters are definitely enzymes. They hydrolyze ATP. The only thing that hydrolyzes ATP productively is an enzyme. So for primary, it's unambiguous. For secondary, they do lower energy barriers for solute transport, and they accelerate the reaction. They do convert one solute gradient into another solute gradient by doing the things that enzymes do. They just don't hydrolyze anything. So this is where it gets fuzzy. So this is why, and that's why this is conversation, not an exam. If I asked you a question like this on an exam, I would say, I think all active transporters are enzymes. Tell me why I think that. You would explain your logic. Is that clear? Questions about that? All right. So now we're going to go through some of this. Simple diffusion, which we're not going to be talking about very much. And the fact that the ions, these are the most important solutes for this course, and they are impermeable to membrane. Membranes are impermeable to ions because ions are so hydrophilic. They're surrounded by water. They're hydrated. And water and charge really has a high energy barrier for going through a non-polar solvent. Here is a real simple view of a cell from the perspective of the sodium-potassium ATPase and from the perspective of sodium and potassium gradients. So you know that the sodium-potassium ATPase hydrolyzes ATP, pumps sodium out and potassium in. In Cell Phys, I didn't need you to know the numbers, but now we're in A&P and I do. It pumps 3 sodiums out for every 2 potassium in. So every time ATP is hydrolyzed by this protein, it's the net flow of ions outward, right? One charge is moved outward. That fact by itself means that the activity of this, the ability of this protein to pump is affected not only by the presence of ATP, and not only by the presence of its solutes, but by the voltage of the cell, because it is trying to pump solutes out. And so if the cell is highly negative on the inside, just the voltage is highly negative on the inside, that is a energy that this pump would have to overcome, in addition to the gradient. In other words, this pump both creates and is sensitive to electrochemical gradients. Here are the concentrations that this pump and other transporters typically maintain across a typical cell. You're going to work with these numbers in a problem set, which I'm not giving you now, but I will give you on Tuesday. These numbers are not dogma. This is typical. Every cell in your body probably has something similar to, but not exactly equal to these numbers. So the typical extracellular sodium is 140 to 150 millimole. The typical extracellular potassium is, this is actually a little high, 3 to 5 is normal. I don't expect you to memorize the details, but I do expect you to know that the sodium concentration on the outside is something above 100, and it's a lot higher than the potassium concentration on the outside. And the potassium concentration on the inside is something higher than 100, 150, 140, 130, depending on the cell. And the sodium concentration on the inside is lower than that by a factor of 10, 12, 15 millimolar. So knowing the exact number is not necessary, but knowing the number within a factor of about 30% is important. It will help you. I'll never ask you a question, what is a typical extracellular sodium? But I might ask you to use that in solving a problem. So knowing it is to your advantage. The sodium-potassium ATPase has a mechanism that pumps. So there's a movie. I forgot to load this up on. Let's watch the animation. Luckily, the sound doesn't matter much. Let me stop. Can you hear that? Anybody hear that? No. What he just said is transmembrane protein. So I'll stop and narrate it every few seconds because I forget. When I use the internet to connect to, when I connect wirelessly, I can't connect sound. So I forgot that. Membrane protein. We're going to go through a cycle. This is the inside of the cell, and this is the outside of the cell. So we always need to get oriented. 3 sodium ions bind to sodium binding sites on the protein, accessing the protein from the inside. Sodium binding initiates a small conformational change, revealing an ATP binding site on the intracellular side. That ATP binding site is in fact phosphorylated. So ATP is hydrolyzed by the protein, not by a kinase that's working with this protein, but by this protein, and it becomes phosphorylated. So ATP binds and a phosphate is transferred from ATP to the protein. That's phosphorylation. So of course, if ADP now is released. Phosphorylation triggers a large global conformational change. It went from inward facing for the sodium binding sites to now outward facing. That was a big conformational change, and it lowers the affinity of those binding sites for sodium. So now sodium is no longer bound with high affinity, and it is outward facing, so you can predict what's going to happen. The internet's even giving you a chance to predict what's going to happen. dissociation. So sodium now has been transported, 3 of them, from inside to outside, and ATP has been hydrolyzed. We're not done yet, but we have transported sodium. The new conformation has a high affinity for potassium ions. It's outward facing, it has low affinity for sodium, high affinity for potassium. So, you know what's going to happen? Binding. Binding of potassium initiates another conformational change. You may have noticed it went by pretty quickly. The binding of potassium initiated a conformational change in which the phosphate that was bound here, phosphorylated, dissociates, and the protein undergoes another large conformational change from outward facing to inward facing. Okay, that's a conformational change. You can predict what's going to happen next. Low affinity for potassium in this conformation. Potassium dissociates. We're now back to where we started, with low affinity potassium binding sites and high affinity sodium binding sites facing inward. And we can do the cycle again as long as there's ATP and sodium and potassium around. So I apologize for the sound. That was a little distracting, but I'll try to remember in the future because I just have to do a different connection and then I can broadcast sound in this room. So in your PowerPoint, this goes through it stepwise. It's upside down relative to the way you just saw it. So this is the inside and this is the outside. So you just have to get oriented for every time you're looking at an image. Here's sodium binding from the inside, ATP binding, and ATP hydrolysis, conformational change, dissociation, binding of potassium, conformational change, or binding of potassium, dephosphorylation, big conformational change, back to the original state. So the textbook does a pretty good job of walking you through the steps. So you can either watch the movie with the animation, with the narration. The link is on, just cut and paste this out of the PowerPoint into your browser and you can listen to you can actually hear the animation. Or you can just read it, or you can just walk through it and narrate it yourself. Okay, yeah, remember, this is my reminder. So, in this course, we're going to see proteins as blobs. We're going to see a lot of globular diagrams of protein. Well, you know better than that. You know that proteins are not blobs. They are three-dimensional structures with and there's a lot of information about protein structure. This is what the sodium-potassium ATPase looks like. This is not what the sodium-potassium ATPase looks like. But this is helpful from a physiological or a functional perspective. But this is what it actually is. And I'm going to try to not beat this to the ground, but I do want to emphasize that we know some of these structures. And so here's the protein structure. And we're not going to redo Cell Phys. I'm not going to ask you about alpha helices and beta sheets. But you should be remembering that every time you see a blobogram, what you're really seeing is this, or something like this. And I'm not giving you the details, except where I need to. Let's take a break for a few minutes. And what we're going to come back and do is calcium transport. I'm going to do this on the board instead of on slides. And then, I forget, something else coming up as well. So let's take a short break and get ready to do calcium transport. calcium gradients, not sodium and potassium anymore. So here's the world's stupidest diagram of a cell, right? You know that there's lots more, I mean, remember cell physiology and CGI even. We've got a nucleus, we've got endoplasmic reticulum, we've got ribosomes all over the place. It's, cytoplasm is a gel, not a fluid. I mean, it's got all kinds of stuff, but here's our cell. What is this? Can you see it like better than this one? What kind of protein is this? Giving you enough hints to tell me the answer to that. Somebody. A primary active transport. Oh, actually, no, you had to know this in order to actually be able to completely answer that question. This is a primary active transporter. It pumps calcium out. It's got a name. So just like the sodium pump has a name, the sodium potassium ATPase, this protein has a name. It's called the plasma membrane calcium ATPase. And it pumps calcium out. So now, knowing that we've named a protein for the membrane that it lives in, and knowing that cells have an endoplasmic reticulum, I want to introduce another here's the endoplasmic reticulum, the world's simplest, dumbest version of the ER. Here is a primary active transporter. That pumps calcium from the cytoplasm into the lumen of the ER. This protein also has a name. Endoplasmic reticulum calcium ATPase. So why isn't it called ERCA instead of, what is the S? The S calls for sarcoplasmic endoplasmic reticulum calcium ATPase. The sarcoplasm is a specialized endoplasmic reticulum in muscle cells. So it gets its own, it gets a letter. I didn't name it, it's what the world named it. The sarcoplasmic endoplasmic reticulum. It really should be sarcoplasmic endoplasmic because those are different words. But it's not, everybody just calls it SERCA. Sarcoplasmic endoplasmic reticulum and calcium ATPase. It pumps calcium into the lumen of the ER. So now here's a key point. The calcium concentration in the cytoplasm is very small. It's on the order of 100 nanomolar, 10 to the minus 9. So that's 0.1 micromolar, or 1 times 10 to the minus 4th millimolar, whatever you're comfortable with. But you need to know that that the calcium concentration in the cytoplasm is very low. And the calcium concentration out here is something around 1 to 2 millimolar. And the concentration in here is something like 10 millimolar. And you don't need to know those numbers, but you do need to know that they're orders of magnitude higher than what's inside the cytoplasm. Orders of magnitude. Like 4 orders of magnitude. 5. That's a big group. Those are the primary active transporters that move calcium and create that very large calcium gradient. In addition, what kind of protein is that? What kind of protein is that? Joey?
Secondary
That's
active.
a secondary active transporter. It's using the sodium gradient that you know all about from the previous slide to pump calcium out. So these 3 proteins pumping calcium out of the cytoplasm establish this very low cytoplasmic concentration of calcium at rest. We're going to use that gradient a lot during this semester. So, this protein has a name. It's called the sodium-calcium exchanger. The sodium-calcium exchanger. Oh, no, that's not what it is. Sodium-calcium exchanger. NCX is the abbreviation for the sodium-calcium exchanger. Now, so in your PowerPoint, you don't have this information, but you do have a reminder that we talked about these proteins in class. So let's talk about another type of sort of combined primary and secondary active transport that we're going to need to know. What's that? Say again? A terminal bouton, a presynaptic terminal? Yeah, excellent. I mean, that with very few clues, you got the reminder that this is an axon going into this, a presynaptic terminal, or a synaptic button, or a synaptic bouton, or any of those names. So, again, we're going to make this really pretty simple. What are those supposed to be cartooning? Synaptic vesicles. What do you know is inside synaptic vesicles? Neurotransmitters. Now we're going to talk about how those neurotransmitters got there. scale is bad. The type of protein, primary active transport, I don't need to put the ATP and the ADP. What is transported? Protons are pumped by this proton ATPase into the lumen of synaptic vesicles, establishing a acidic inside and a proton gradient. Higher concentration of protons, low pH, inside the synaptic vesicles compared to the cytoplasm of the presynaptic terminal. Okay, well, we haven't gotten neurotransmitter there yet, but you can probably imagine what this is going to be. Protein, secondary active transport, using the proton gradient, proton going down its concentration gradient in exchange for some neurotransmitter, specific for the different actual neurotransmitter, but we're just going to stay generic now, so specific for glutamate or GABA or acetylcholine or whatever. But for now, let's just call it neurotransmitter, pumping neurotransmitter into the lumen of the vesicles. How did the neurotransmitter get into the cytoplasm in the first place? Well, it was either synthesized there, or it may have been re-uptaked after, I'm jumping ahead, just to complete the story a little bit. You'll remember that neurotransmitter is exocytosed during chemical neurotransmission. We'll get to that again on Tuesday. But just how this neurotransmitter is then re-uptake from the synaptic cleft in a sodium, you can't see over there. I'll do it up here as well. Can you see it if I do it up here? Neurotransmitter is retaken up after exocytosis by a sodium neurotransmitter co-transporter. We're going to see this again. The concept for today is to remember primary active transport coupled with secondary active transport can create the gradients you really need. Pumping neurotransmitter into the presynaptic terminal and then pumping it from the cytoplasm of the presynaptic terminal into the vesicle. One of them is a sodium co-transport and the other is a proton exchanger or antiport. Joey?
So the one on the bottom is just, you just rewrote it up on the top?
Say again?
The blue one on the bottom is the same thing, you just wrote it up on top?
Yes, but what I meant to do is
You
do
didn't- Okay.
neurotransmitter. This and this are the same thing.
Okay. You just said it twice, so I was just trying to make
I was
sure
just
that-
doing this up here because they couldn't see this one back there. I'm being a little dishonest about where these things are. These are down here. So now, to sort of finish up for today, I just want to reinforce a key point. And that is this blobogram, right? So here is the cartoon version of a uniporter. The cartoon version is a glucose uniporter. which is important in neurons because it's how glucose as an energy source gets from the blood, from your body, into your neurons. Just as an aside, your brain doesn't have energy stores. It's unlike liver and muscle that can survive for a while on its own intrinsic energy source. The brain is constantly dependent on blood. So you deprive blood flow to a brain cell and it's dead in a few minutes. So, and that's because it's requiring glucose from blood, from your body, from your recent meal, or from your liver if you're fasting, or from your muscles if you're fasting. It's requiring glucose from blood, and it's getting from the outside to the inside by a uniporter. Passive transport. Here's the blobogram. Here's what it actually looks like. This isn't, here is the protein it's mostly alpha helical. Here are cartoons of glucose or other sugars that are bound to a binding site. The membrane, so here, this is just showing the protein. The membrane is probably from here to here. This part of the protein is embedded in the membrane. That's what it really looks like. This is what you're going to be seeing a lot. Channels. Conformational changes from open to closed and closed to open. We're going to talk a lot about those conformational changes. Blobogram, a potassium channel, what it actually looks like. So here is, actually this is a sodium channel, sorry. Not a potassium channel, a voltage-gated sodium channel. And we're going to be talking about some of these domains of the channel. So this is looking at it from the side. This sort of gray vase-like structure is the pore. It's showing the hole down the inside of this protein. In the case of channels, we might actually get into some specific molecular physiology. We'll see. Again, I guess I am beating this to death. The structure of an exchanger, the sodium-calcium exchanger. Can anyone guess why there are really, so in each of these images, there are two colors. What's the difference between the two colors? What do you guess? Knowing what you know about the sodium-calcium exchanger, what is your guess as to why they showed two colors? Sophie? One is in the sodium-bound state, and the other is in the calcium-bound state, the two different conformational states of this protein. In the blobogram, it goes back and forth like this. Well, it's not that big a deal, but it's still a large conformational change. And you can see, like for example, especially this helix right here, undergoes a large conformational change from the sodium-bound state to the calcium-bound state. That's those are the conformational changes. Can anyone guess why this and this are shown? They look almost exactly the same. Why would a paper show two figures that for all practical purposes are the same? This is incredibly cool. This is one of those stereograms. If you can learn how to defocus your eyes and superimpose these two images together and make a three-dimensional image. I'm sure I couldn't do it on the screen either, but when you have it on paper, try either crossing your eyes or defocusing your eyes and making a third image and get it in focus, and it will be three-dimensional. It's fun. So here is a textbook cartoon of the different transporters we're going to be talking about. We've talked about them already. The sodium potassium ATPase. 2 different calcium ATPases, one in the plasma membrane and one in the sarcoplasmic endoplasmic reticulum. A proton ATPase, the one we're going to be talking about mostly is the one in the synaptic vesicle membrane. Exchangers, sodium, well, secondary active transport. This is a co-transport. We're not going to be talking too much about amino acids except for when they are neurotransmitters. Sodium neurotransmitter, co-transport. Sodium calcium exchange. Those are the ones we've talked about so far. I don't think we'll talk about the others too much. Glucose, to get the source of energy into your brain cells as a unit form. And we're going to talk a lot about ion channels. Sodium channels, potassium channels, calcium channels, and to a lesser extent, chloride channels. Okay, AMP. Welcome. I see some of you this evening.